It is a great honor and pleasure for me to be here in Japan, the home of my doppelganger. Dr. Gero is a character in the comic Dragon Balls, and we share more than just the first name. He's also a scientist, a mad one, who strives for world domination, just like I do. And if you look very carefully, you can see that his skull has been replaced with a transparent plexiglass dome, so that the working of its brain can be controlled with light. This is done by modifying specific types of brain cells genetically so that they express light-sensitive membrane proteins that transduce optical commands into electrical signals. And these two essential ingredients, optics and genetics, have given the method that's being recognized by this year's JapanPrize its name, Optogenetics. Now, what motivated the invention of optogenetics was the idea that a methodology like this would open 3 experimental doors for neuroscience that had previously been locked. The first was an understanding of the neuronal causes that underpin our experience. The idea was that if one could insert artificial test patterns into the nervous system, ask the brain to decode them for us, and fractionate them according to their power to evoke perception, action, emotion, or thought, one could gain insight into the fundamentals of brain operation that were previously not possible. The second locked door was the exploration of neuronal connections, which is traditionally done painstakingly with pairwise surges with electrodes. If one could replace one of these electrons with a light beam that elicits activity in just a subclass of genetically targeted brain cells and records responses in other connected partners, that would increase both the throughput and the resolution of the method dramatically. And finally, optogenetic control would give us an opportunity to explore neuronal mechanisms. As every scientist knows, if one has a hypothesis about how a biological system works, the only way to find out whether that hypothesis is right or wrong is through targeted perturbation. So for most of my lecture today, I will focus on our studies of one biological problem in which optogenetics has indeed opened all 3 of these previously locked doors for us and allowed insights into a mysterious biological process. The problem I'm talking about is the neuronal function and control of sleep. Sleep is one of the great biological mysteries. Each night we disconnect ourselves from the world for 7 or 8 hours, a state that leaves us vulnerable and unproductive. And despite these risks and costs, we still don't know what sleep is good for. We only know that it must be good for something really essential, because no animal has evolved that can bypass the need for sleep. Now, we try to solve this problem by understanding the brain processes that regulate sleep and waking. And there's in fact 2 of these brain processes that are symbolized by 2 different forms of oscillation on this slide. The sine wave symbolizes the circadian clock, which oscillates in synchrony with periodic changes caused by the Earth's rotation. As such, it is purely an adaptive mechanism that makes sure we do our sleeping at the right time, at the time that suits us best, but it cannot speak to the deeper problem of why we need to sleep in the first place. The answer to this problem, we think, will come from understanding of the second control mechanism, the sleep homeostat, that is symbolized here by the sawtooth oscillation that's superimposed on the sine wave. The homeostat measures something, and we don't know what that something is, that accumulates or depletes during waking. And when that process reaches a certain threshold, the mechanism puts us to sleep. The accumulated sleep pressure is discharged during sleep, and the cycle begins anew when we wake up. Now we understand a great amount about the function of the circadian clock. And this paper here is really the Rosetta Stone that broke the problem open. The discovery by Seymour Benza and his graduate student Ron Konopka more than 50 years ago of fruit flies whose circadian clocks ran abnormally fast or slow. And from that discovery then flowed, through the work of many laboratories, a pretty complete molecular, cellular, and systems understanding of circadian timekeeping. This slide, in contrast, and it's an overstatement, but probably not too gross an overstatement, summarizes much of what we currently know about the molecular function of the sleep homeostat. My goal for the rest of this lecture is to draw at least some outlines on this blank canvas. Now, we know conceptually what the sleep homeostat must do. It must be what an engineer would call a relaxation oscillator, a system that can exist in two states, a filled state and a discharged state, that alternates between these states, and in one state accumulates a process that runs during waking and increases the pressure to sleep, and that process is then discharged during sleeping. Now, when I said before that we know virtually nothing about sleep homeostasis, that of course was incorrect. We do know quite a bit about the neuronal substrates and the brain regions that are involved in homeostatic sleep regulation, but what's missing is a molecular understanding of sleep homeostasis that would be equivalent to the understanding of the translational feedback oscillator that controls circadian timing. Now, the early insight, the Rosetta Stone in this case, actually came during the last great pandemic that swept the world. Toward the end of World War I, a young neurologist in Vienna, Konstantin von Economo, described patients with a very unusual group of sleep disturbances. Some of these patients were somnolent or lethargic. So von Economo called this disease encephalitis lethargica. But a minority of patients, as the disease progressed into its chronic stage, actually became incurable insomniacs. And as these survivors of encephalitis lethargica began to die off, Economo performed autopsies that confirmed very perceptive clinical observations that suggested that a particular area in the brainstem or the hypothalamus is responsible for the active induction of sleep. The important conceptual consequence of this discovery was that sleep is not just a passive process of the brain shutting itself off in the absence of external stimulation or internal arousal, but is actually a process that must be actively induced and maintained. Now, what we are approaching is the study of the equivalent set of neurons in the brain of fruit flies. So we are picking up the thread of Benza and Konopka, who understood circadian timekeeping by studying the circadian rhythms of fruit flies, whereas we have been studying sleep homeostasis by exploring the biophysics that regulate the activity of the fly's equivalent of von Economo's sleep control neurons in humans. You see these neurons here labeled with the green fluorescent protein. They are, as in humans, a small minority of all neurons in the brain, about 24. The axons project to this inverted V-like structure in the midline, and their cell bodies lie to the right. The axonal projections target a substructure of the central complex called the dorsal fan-shaped body. And these neurons here are labeled with a genetic control element called R23 at E10. So whenever you see this combination of numbers and letters, you know that we target a genetic manipulation specifically to these classes of neurons. Now, the sleep-inducing capability of these cells was revealed early on, shortly after their discovery by a former postdoc in the lab, Jeff Donnelly, while he was actually a graduate student with Jeff Shaw at Washington University in St. Louis, of the sleep-inducing capability of these cells. What you see here is the sleep profiles of flies over the course of 2 days. These flies are not showing a pronounced circadian rhythm because the experiment is performed in complete darkness in order not to provide an optogenetic stimulus at the inappropriate time. So you can see that control flies and flies that express our optogenetic actuator, a light-gated ion channel, just in these sleep control neurons, sleep similarly during day 1. But during the subjective day period of the second stimulation day, we switch on the lights, and you can see that the light itself has a slight wake-promoting effect, because the control flies sleep slightly less and then rebound at 12 hours after we switch the lights off, whereas the flies that express the light-gated ion channel in the sleep-inducing neurons go to sleep instantly and stay asleep for the entire duration of the stimulation period. Now, studies of these sleep control neurons in many different flies revealed that these cells are biophysically very unusual in that they switch between two states whose extremes are shown here. One state is the state that you would normally expect from a well-behaved nerve cell, shown here on the left. You see that these neurons respond to the injections of depolarizing currents with action potentials. You see these as these little spikes that ride on the membrane voltage signal, the membrane potential signal. And the frequency of these spikes increases with the amount of injected depolarizing current. Whereas the sleep control neuron on the right does not emit any action potentials at all, even when it's step depolarized to positive membrane voltages. And in addition, you can see that the same amount of current injection causes much lower voltage changes, meaning that the neuron on the right opposes the injected current with a lower resistance than the neuron on the left. It also settles into its new membrane potential equilibrium much faster than the neuron on the left. And this combination of low input resistance and what's called a short membrane time constant is characteristic of the opening of a current shunt, whose molecular basis I'll show you in a second. So when we saw this, we of course thought, well, perhaps this is the mechanism of homeostatic sleep control, that these neurons, depending on sleep pressure, actually switch between electrically active and electrically silent states, and it's this state switching that underpins the transitions between sleep and wakefulness. And indeed, when we manipulated the sleep histories of flies, we found more neurons in the electrically active state on the left in flies whose sleep pressure was high, that is, flies that hadn't slept for a long time, whereas in well-rested flies, shown on the right, most neurons were electrically silent. But of course, in order to make this correlative evidence causal and show that the sleep-inducing neurons indeed switch as a function of sleep pressure between these two states, one would like to be able to induce such a state switch directly, on command. And in order to do this, of course, one would want to know a signal that normally operates this switch. Now, what might such a signal be? A hint had come from the first experiments in which the behavior of an animal was controlled optogenetically. These experiments were performed in the year 2004 by my then graduate student Susanna Lima at Yale University and published in 2005. What Susanna had done was she had expressed a light-gated ion channel in all neurons in the fly's brain that communicate through the neurotransmitter dopamine. And then she had recorded the locomotor patterns of these flies over a course of 2 minutes in a circular arena, before and after switching on the dopaminergic system with light. And you can see here the same flies before and after this optogenetic intervention. And it should be obvious from comparing the locomotor trajectories before and after that dopamine is highly, highly arousing. The flies become much more active in the bottom row than they were in the top row. Now, dopamine is, of course, an arousing neuromodulator, not only in flies, but also in humans. This is how psychostimulant drugs like cocaine and amphetamine work. They block the reuptake of dopamine from the synapse into the presynaptic terminal. So if dopamine is indeed an arousal-promoting substance in the fly, one would expect to find a dopamine delivery system that actually targets the sleep-inducing neurons, and that is the case. If you look at the same brain region that's inhabited by both classes of neurons, you can see that their projections shadow each other very, very closely. So this naturally raises the question, are these two types of neuron actually connected? And optogenetics once again gives us the perfect tool to answer this question. We can express a light-gated ion channel in the putative presynaptic partner, in our case the dopaminergic neuron, and record from the sleep-inducing neuron and see whether dopamine, the arousing signal, can actually switch the sleep-inducing neuron from the electrically active to the electrically quiescent state. So here's such an experiment where we now record from a sleep control cell. It starts out in the electrically active on state. Then we deliver dopamine optogenetically for a short period of time. And you can see that the cell undergoes the complete state switch. It ceases to emit spikes or action potentials. And it also shows the passive membrane changes, the drop in input resistance, the shortening of the membrane time constants that we have seen before in the recordings from flies whose sleep pressures were high and low. Importantly, if we hold the recording long enough and stop the dopamine delivery, we occasionally see that the sleep control neuron spontaneously reverts to the electrically active on state. So this is not an artifact that's induced by our experimental conditions, but part of the normal duty cycle of these neurons. Now, the mechanism that underlies these biophysical changes in the electrical properties of these neurons, I'll summarize in just a few slides. We know, or we found that early on, that potassium currents are the most likely dominant contributors to these electrical excitability changes. And potassium currents typically are divided into two broad categories: voltage-dependent, so-called A-type currents, and voltage-independent, so-called non-A-type currents. You can see here that the A-type current is high in the electrically active on state and drops when dopamine switches the neurons off. So it's high, the A-type current is high during sleep and low during waking, and returns to an elevated level upon waking up again. Whereas the non-A-type current shows the exact opposite behavior. It is typically suppressed during sleep, becomes the dominant potassium current in the off state during waking, and is once again suppressed during sleep. We also found out through RNA-mediated interference what the molecular carriers of these potassium currents are. The A-type current is mediated by the prototypical voltage-gated potassium channel called shaker, whereas the non-A-type current flows through a two-pore domain leak-type potassium current that we've called Sandman. So what we have achieved through this biophysical analysis of a set of neurons that are causally important for the induction of sleep is that we have reduced a complex and vague problem that is difficult to address experimentally, namely, what is the function of sleep, to a problem that we can actually attack experimentally. We can replace this vague question with the question of what regulates Shaker and Sandman. Then it's the regulation of these two ionic currents that is responsible for the regulation of the biophysical properties of the sleep control neurons, which in turn are responsible for the induction of sleep and waking. I will focus for the rest of my talk on the Shaker current only, because there the progress has been most substantial. We are working with great intensity also on understanding Sandman. This is a beautiful molecular structure of the Shaker current of mammals, showing that it consists of two portions: a membrane-spanning ion-conducting alpha subunit shown here in gray, and appended to this alpha subunit shown here in blue is a beta subunit that hangs sort of like the gondola under a hot air balloon. Now, these beta subunits have puzzled molecular biologists, neurobiologists, ion channel biophysicists since the first sequences and then the structures were determined in the early to late 1990s. Because it was found that these proteins resemble enzymes, specifically oxidoreductases or aldoketoreductases. And you can see in the active site, there's actually a cofactor bound, the molecule here in red, which is called NADPH. So seeing these homologies between these ion channel beta subunits and enzymes raised the question of what the function of this particular association in the ion channel complex is. Is this whole assembly a voltage-controlled enzyme, a voltage-controlled aldo-keto reductase, or is it a redox-controlled ion channel? I will show you evidence that it's both, and it is actually the dialogue between the conducting pore of the channel and the active site of the enzyme that's the fundamental accounting mechanism that measures sleep pressure or elapsed waking time. The model, again, I will summarize just in a few slides because most of this work is published, is that the shaker channel beta subunit, which in flies is called hyperkinetic, actually senses through the oxidation state of its cofactor NADPH how efficiently the mitochondrial electron transport chain burns fuel to create energy for the nerve cells. What you see here is a mitochondrial electron transport chain where the supply of electrons from the metabolism, mostly of carbohydrates, but also ketone bodies and fatty acids, is in balance with the demands of ATP synthesis, which are fulfilled by the turbine that you see spinning on the right. So each electron that enters the chain at complex 1 flows down the chain smoothly, pumps protons into the intermembrane space, and these protons then flow down their gradient and power the ATP synthesis, the turbine on the right. But if the amount of electrons entering the chain is in excess of what the ATP synthase can use, these electrons begin to back up in the transport chain, spill from the mitochondrial transport complexes, especially complex 1 and complex 3, and transfer directly to molecular oxygen in a disorderly fashion. They create what's called reactive oxygen species, primarily superoxide, that then gives rise to a variety of breakdown products. And we have shown that it is this production of reactive oxygen species in the mitochondria that is somehow sensed by the shaker hyperkinetic complex in the plasma membrane, which changes the oxidation state of the cofactor from NADPH to NADP+. And this conversion is sort of an elementary bit that contributes to the accumulation of sleep pressure during waking. I'll show you just two key pieces of evidence that support this model. The first one focuses on the mitochondrial electron transport chain, which we see here in its backed up situation where an excess of electrons is entering at complex 1. It turns out that many plants and some microorganisms have a relief valve, an alternative oxidase called AOX, which takes these surplus electrons and transfers them safely to water. So this AOX taps into the ubiquinone pool and is like a pressure relief valve that prevents the electron transport chain from overheating. Now, when we installed this alternative oxidase into the electron transport chain of the mitochondria of the fly sleep control neurons, we could cut total daily sleep by about 50%. So installing this overflow valve in the electron transport chain dramatically relieved the pressure to sleep. On the other hand, the focus on the evidence, the clearest evidence implicating that the potassium channel beta subunit is the crucial sensor of how efficiently the mitochondrial transport chain burns fuel, comes from experiments where we once again used an optogenetic trick to influence the redox state of the cofactor directly. That trick relies on a protein called Minisog, which was developed by the late Roger Chen as a contrast agent for electron microscopy. And upon illumination, this Minisog produces a different kind of reactive oxygen species called singlet oxygen. So Minisog is just an abbreviation for miniature singlet oxygen generator. We anchored this Minisog protein at the plasma membrane, again, of our sleep control neurons in the intact fly next to the shaker potassium channel and its hyperkinetic beta subunit, and then illuminated the intact flies. And you can see that a 9-minute period of illumination is capable of putting flies that express minisorg in the sleep control neurons to sleep, both in greater number and for longer periods of time than control flies. And this sleep-inducing activity requires hyperkinetic as the crucial transducer, because in the very column on the right, you can see that we have removed the beta subunit hyperkinetic through cell-specific RNA-mediated interference with its expression, and that abolishes the sleep-inducing effect completely. Now, the ability to switch the redox state of the beta subunit on demand by optogenetic intervention also gave us an opportunity to actually study the biophysics of these neurons by once again recording their electrical properties, then illuminating them and seeing what changes. And the biophysical mechanism that underlies the increase in sleep-inducing electrical output that you see here on the left after illumination and summarized in the middle diagram as these current spike frequency curves, which get steepened through the illumination, is a slowing down of the inactivation time constant of the potassium current, which you see on the right. The intuition that couples the inactivation kinetics of the potassium conductance to the spike frequency is that with each action potential, you would normally push a certain percentage of all potassium channels into the inactivated state, and that slows the firing frequency because you reduce the repolarizing force that pulls the membrane potential back to its resting level after each action potential. But if you slow down inactivation, you reduce the percentage that becomes inactivated, and that means that the neuron can sustain its sleep-inducing activity at a higher frequency for longer. So in this way, mitochondrial electron transport through this mechanism is coupled to heightened electrical activity of the sleep-inducing neurons. But there was one thing that sort of bothered us about this model, and that is the considerable distance that these reactive oxygen species that are produced in the mitochondria would have, and that are very, very reactive, would have to travel to reach Kv beta with its bound NADPH at the plasma membrane. So we thought that perhaps there is a crucial intermediate that is initially attacked by these reactive oxygen species, and that crucial intermediate is of course an abundant and highly vulnerable substance to oxidative damage, and these are the polyunsaturated fatty acids of membrane lipids. This is the classic lipid peroxidation chain reaction studied by chemists, where a radical such as the hydroxyl radical shown on the lower left reacts with the bisallylic carbon of a polyunsaturated fatty acid to form a lipid radical that then, under the consumption of oxygen, forms a lipid peroxyl radical, which then attacks another polyunsaturated fatty acid and propagates the chain while simultaneously producing a lipid hydroperoxide. And indeed, we performed in collaboration with the group of Bernhard Spengler a study of how the brain's lipid composition changes after sleep deprivation by performing mass spectrometric imaging of cryosections. And you can see that there's dramatic changes in specific phospholipid species that are either enriched in rested or in sleep-deprived substances. In fact, these changes are so characteristic that we can, with perfect accuracy, distinguish rested and sleep-deprived brains from their lipid composition alone. Now, what underlies this perfect discriminability of the two physiological states based on their lipid profiles is, in fact, a number of changes that affect both the head groups and the fatty acyl chains. But the most crucial change, as you can see, if you look and count the number of double bonds on the left in the rested state, the average is about 5 double bonds per phospholipid molecule, whereas in the sleep-deprived state on the right, the average is a little more than 1, between 1 and 2. So that means that polyunsaturated fatty acids with a high number of double bonds get selectively depleted during sleep deprivation, presumably because they are vulnerable to oxidative attack, and during waking these lipids are slowly depleted. Now, interestingly, this lipid peroxidation chain reaction, which produces the lipid hydroperoxides that are shown here on the right, actually has end products because these lipid hydroperoxides undergo a series of fragmentation and scission reactions that actually then produce carbonyls, aldehydes, and ketones such as 4-oxo-2-nonenal, which are, of course, the perfect potential substrates for an aldo-keto reductase such as the beta subunits. They have exactly the right chemistry. So we then decided, of course, to test whether this model that the mitochondrially derived reactive oxygen species initially attack polyunsaturated fatty acids of membrane lipids, which then fragment into lipid-derived carbonyls, which are the substrates for the beta subunit, that reduces them to alcohols, and this reduction is coupled to the oxidation of the cofactor NADPH to NADP+. We tested this model by including in our patch pipette 4-oxo-2-nonenal and using the inactivation time constant of the potassium current as our measure of whether the ion channel is predominantly in the NADPH bound or reduced, or the NADP+ bound or oxidized state. And you can see that if we add our model aldehyde, we can indeed shift the channel population rapidly within 10 minutes to the oxidized state, the NADP+ bound state. This shift requires catalytically competent beta subunit. If we use a mutant that has a difference of only one amino acid that breaks this mechanism, the increase in the inactivation time constant does not occur. Importantly, the increase is also stable for a very, very long time, suggesting that the enzyme has stop-and-go kinetics, that it doesn't turn over continuously, but that the cycle arrests after one oxidation step and NADP+ stays stably bound. A more powerful way of demonstrating these stop-and-go kinetics is actually to use the optogenetic mini-SOG trick and illuminating mini-SOG, using this to shift the channel population to the NADP+ bound state and then to switch the lights off. Whatever chemical product is produced is so short-lived, the longest half-lives are on the order of 3 to 4 seconds, that over an extended period such as 20 minutes, if the enzyme did turn over, the NADP+ should have been replaced by NADPH, but as you can see here, it is clearly not. This molecular device, in fact, acts like a biochemical memory for a single oxidation event that in its totality, meaning in the totality of the hyperkinetic pool of the sleep-inducing neurons, quantizes the analog variable of sleep pressure, elapsed waiting times, into the discrete binary states of many of these beta subunits of the ion channels. Now, if this is really the physical measure of sleep pressure, then one would expect that it would be dissipated during sleep. And the crucial change that occurs during sleep in these sleep-inducing neurons is, of course, that they increase their electrical output, they increase their firing rate. And as you can see, if we simulate this sleep-inducing electrical output of the neurons, we can erase the biochemical memory that has accumulated. In other words, irrespective of whether we have changed the oxidation state of the cofactor chemically with our model compound or optogenetically with mini-SOG, the increase of the inactivation time constant is reversed. In other words, NADP+ is expelled from its binding site and replaced with NADPH, which is about 40 to 50 more abundant in the cytoplasm. So what we have then here is a biochemical memory cell that in many respects resembles the semiconductor memory that's used in our computers or cell phones. A dynamic random access memory cell has also two components, just like the potassium channel. It has a storage capacitor that holds information, a single bit, in the form of an electrical charge. And whether that charge is written or read, whether that charge is altered, is determined by the voltage across an access transistor. And the whole assembly has two address lines, one called a word line and the other one a bit line. The word line determines the voltage across the access transistor. The bit line determines what is written to the storage capacitor or read from the storage capacitor. The analogy in our case, we think, is that the beta subunit is the equivalent of the storage capacitor, but information is not stored in the form of an electrical charge, but in the form of the oxidation state of NADPH or NADP+, so a single bit. And the alpha subunit, which is voltage sensitive, plays the role of the access transistor. The two address lines at whose intersection the memory cell sits, in our case, have one electrical input, which is the membrane potential of the sleep control cell, and one metabolic input, which is the efficiency with which the mitochondrial electron transport chain operates. So we've reached a stage, I think, where we can replace this conceptual animation of how we think sleep homeostasis operates with a molecular animation. This is a picture of sleep-inducing neurons with these two crucial potassium channels, Sandman, that I haven't talked about at all, shown here in yellow, which we've demonstrated cycles between intracellular storage vesicles and the plasma membrane, and is the principal determinant of whether the neuron is in the electrically active on or the electrically silent off state. When the neuron is in the off state, in other words, during waking, the beta subunit of the voltage-gated potassium channel, which is the blue protein that sort of hangs like the gondola underneath the pink ion channel, progressively fills with NADP+, because the wake state is associated with higher levels of oxidative stress due to higher metabolic rates, either of these neurons or in all likelihood brain-wide. Once the Sandman block on the electrical output of these neurons is lifted, the NADP+ can leave its binding site, is replaced by NADPH, and the entire cycle begins anew. So what this then really suggests is that perhaps an ancient fundamental function of sleep is to protect cells with high metabolic rates, cells that are non-renewable, and extensive membrane systems, almost a definition of neurons, from the damage that they would incur during oxidative stress. And there is at least correlative evidence that suggests that this is true. There is empirical scaling relationships between the amount of daily sleep that mammalian species need and their mass-specific metabolic rate. In other words, how much energy these cells burn on a weight basis or on a mass basis. And you can see that the smallest known mammals, such as the tree shrew, which weigh less than a gram, spend most of their existences asleep, whereas elephants or other large mammals have sleep requirements of maybe 2 or 3 hours a day. The common denominator between these changes and another striking change, namely differences in aging rate and life expectancy, which are very short for small mammals and quite long for large mammals, is these differences in metabolic rate. In other words, small mammals run their metabolism at a much hotter rate than large mammals do, and this may explain these differences. Before I conclude, I would like to take you back just for 1 minute or two into the earliest days of optogenetics, the Stone Age of optogenetics, if you wish. As we began to work on the first of these light-sensitive membrane proteins that are now used to control the electrical activity of excitable cells, I became aware that I was not the only scientist who had seen the need for a method like this. Through a citation alert, I found out that Francis Crick, in an article that was published in the millennial issue of the Royal Society and entitled "The Impact of Molecular Biology on Neuroscience," wrote, "The next requirement for understanding the brain is to be able to turn the firing of one or more types of neuron on or off in a rapid manner in the behaving animal. The ideal signal would be light. This seems rather far-fetched, but it is conceivable that molecular biologists could engineer a particular cell type to be sensitive to light in this way. So when we had the first experiments that turned Crick's far-fetched possibility into a reality, and these are these experiments, these are hippocampal neurons grown in culture and engineered genetically to express a light sensor borrowed from the eye, an opsin protein. You can see that the membrane potential of this neuron hovers around resting level in the dark, but as soon as we switch on the lights, the cell responds with a volley of electrical impulses. So when we had these experiments, I decided to send a preprint of the paper to Crick. And if you've read the wonderful book called The Eighth Day of Creation, you know that Crick was engaged in many different areas of research and took a strong intellectual interest and often also lead in them by steering the development of various fields through a vast correspondence. And his stylistic hallmarks as a letter writer were twofold. He always mixed encouragement and constructive criticism. And this is exactly what I got. Crick replied and said that he had read the paper I had sent him with great interest and was excited to see that the system already works, at least to some extent. However, he realized, as I did, that it still needed improvement and that this will take further work. So this is just encouragement for us to keep on with our efforts. Unfortunately, Crick didn't live to hear how not only our experiments progressed, but also those of many others, most notably the discovery of the molecular identity of channel rhodopsin by Peter Hegemann a little more than a year after this paper was published, and then the first demonstration by Karl Deisseroth of the astounding performance of channel rhodopsin in neurons, about which you will hear later this afternoon. But I think it's fair to say that as a consequence of these collective efforts, the way we neuroscientists approach our work has fundamentally changed. We have matured from being observers to being doers. With that, thank you very much for your attention, and thank you also to the many individuals who have contributed to this work. My current laboratory is shown aligned to the left, and some of the notable alumni whose work I've mentioned have been displaced by one tab stop to the right. Boris Semmelmann was the postdoc who built the first optogenetic actuators and demonstrated their performance in cultured neurons. Susanna Lima was the graduate student who first controlled the behavior of an animal optogenetically. Jeff Donnelly and Diogo Pimentel were the neurons who initially worked out the biophysics of state switching of the sleep control neurons and the involvement of the sleep control neurons in sleep homeostasis. And Anissa Kempf was a postdoc who, together with a former graduate student, Michael Song, discovered the fundamental connection between mitochondrial energy metabolism and the activity of the sleep-inducing neurons and the crucial role of the potassium channel beta subunit, whereas the memory function of the beta subunit was explored by a current graduate student, Olaf Rossmann. Thank you very much.